What if I told you that the largest, most magnificent flower structure on Earth smells exactly like a 3-day-old rotting corpse?
I mean, it's quite the visual.
Right.
Or olfactory experience, really.
Right. And what if I told you that to see it bloom, you have to wait up to 10 years for an event that is entirely over in just 24 hours?
Yeah, it's an incredibly fleeting moment.
So imagine you are standing in the pitch-black undergrowth of the Sumatran rainforest. It's midnight, the air is thick, the humidity is hovering well over 80%, and the heat is just pressing down on your shoulders.
Classic tropical understory.
Exactly. You're surrounded by the ambient noise of a million insects. And then suddenly you are hit by a smell so visceral, so overwhelmingly putrid, that your most basic ancient survival instincts just scream at you to turn around and run.
Oh yeah, it hits the back of your throat.
It smells like a rotting carcass that has been left out in the blazing tropical sun. But here's the catch. You aren't walking toward a dead animal. You're walking toward a botanical masterpiece.
It is a phenomenal image to start with, because it really captures the sheer shock and, the cognitive dissonance of encountering this organism in the wild. You expect a predator or a gruesome scene, and instead you find a plant playing a highly sophisticated trick on the entire ecosystem.
Well, welcome to today's deep dive. Our mission today is to explore the dramatic, the mysterious, and the, frankly, awe-inspiring biology of the ultimate botanical oddity. We're talking about Amorphophallus titanum.
Which most people know as the Titan Arum.
Right. Today, we are going to completely deconstruct how it creates its foul stench, the internal biological furnaces it uses to spread that scent into the jungle canopy, and ultimately, the tragic fragility of its existence in the modern world.
And to do this justice, we are pulling from a remarkably dense stack of recent scientific literature. Rather than just, giving you a reading list, we're going to weave these discoveries together as we go.
Yeah, we have a lot of ground to cover.
We do. We'll be looking at everything from a comprehensive biochemical systematics and ecology paper on the microscopic volatile chemistry of its floral odors to some really fascinating stuff from PNAS Nexus and on the Plant Journal on the molecular basis of how plants generate heat.
Plus Cyril Claudel's exhaustive dissertation on the genus Amorphophallus.
Exactly. And all the way to some exhaustive field research from 2024 and 2025 detailing the desperate fight to save this species in Indonesia. We are treating this plant not just as a static piece of flora, but as an active trickster protagonist in the rainforest ecosystem.
So let's start by addressing a massive misconception that always pops up, because when you say corpse lily or corpse flower to someone, 9 times out of 10, they are picturing the wrong plant entirely.
Oh, absolutely. They picture Rafflesia arnoldii.
Right, which is that wide, flat, bright red, leathery flower that sits directly on the forest floor, like a parasitic pancake.
Yeah, a parasitic pancake is a good way to describe it.
Yeah.
But that is not what we're talking about today. It's a very common mix-up. Both plants smell heavily of rotting meat, and both happen to live in the same general region of the world.
Primarily the Indonesian archipelago, right?
Right. But they are entirely different beasts on an evolutionary level. Amorphophallus titanum is not a lily, and structurally, it's not even a single flower. It's what botanists call an unbranched inflorescence.
Okay, I always get tripped up on that term. An inflorescence essentially means a cluster or a community of tiny flowers masquerading as one giant structure.
That is the perfect way to visualize it. The Titan Arum is essentially a massive, towering architectural structure. You have this central, fleshy, pale yellow spike called the spadix.
The part that shoots straight up into the air like a pillar.
Exactly. And wrapped around the base of that spike is a gigantic, frilly, petal-like leaf called the spathe. It looks almost like an upturned bell or a chalice. And hidden way down at the very bottom of the spadix, deep inside the protective dark chamber created by that wrapped spathe, are hundreds of tiny, individual male and female flowers.
The scale of this structure is just staggering to me. Some of the field data points out that this inflorescence can reach up to 3.7 meters tall.
No, that's over 12 feet.
12 feet.
Yeah.
You're looking at a flower structure that is twice the height of an average human being. But to me, the most fascinating part isn't what is reaching up into the canopy, it's what is happening underground.
Right, the fuel source.
Exactly. To power a 12-foot-tall biological machine, you need a tremendous amount of fuel. The Titan Arum grows from a massive underground corm, which is basically a giant tuber, similar in function to a potato.
Really, really big potato.
Yeah, because this corm can weigh up to 150 kilograms.
Think about the implications of that for a moment. 150 kilograms is over 330 pounds. To put that in human terms for you listening, imagine burying a full-size refrigerator in your backyard.
A refrigerator made entirely of dense, stored botanical energy.
Mostly starches, yeah. The life cycle required to build up that energy is just a masterclass in extreme patience.
Well, walk us through that life cycle, because it really puts the rarity of the bloom into perspective.
So the plant spends anywhere from 5 to 10 years just sitting in the jungle, quietly gathering energy. It throws up a single tree-sized leaf.
Just one leaf.
Just one. And when I say tree size, I mean a single leaf that can be up to 7 meters tall, branching out into leaflets so it looks like an entire canopy of a small tree.
Wow.
Its only job is to photosynthesize, absorb sunlight, and furiously pump sugars down into that underground corm. It does this year after year. The leaf stays up for maybe 12 to 18 months, dies back entirely so the plant goes dormant beneath the soil, and then a new, slightly larger leaf emerges.
And it just repeats this over and over.
It repeats this grueling cycle until it finally hits a critical mass of stored energy.
I love the analogy of someone working a grueling, exhausting corporate job for 10 solid years, hoarding every single penny into a savings account just to blow their entire life savings on one absolutely wild, chaotic 24-hour weekend in Vegas.
That's exactly what it is.
Yeah.
After a decade of silent preparation, it attempts a bloom that only lasts a day or two.
But here is where it gets really interesting. Before it even gets to the flowering stage, before it even attempts to pretend to be a corpse, it's already playing mind games with the ecosystem, right?
Oh, definitely. The historical context of its discovery highlights this perfectly. Cyril Claudel's exhaustive dissertation from 2025 on the genus Amorphophallus details how it was first discovered by Western science in 1878.
By an Italian botanist, Odoardo Beccari.
Right. Beccari was exploring the rainforests of Sumatra. But what is fascinating is that Beccari didn't stumble upon this plant deep in some uncharted, inaccessible, mist-shrouded wilderness.
He didn't.
No, he found it in the immediate surroundings of a local village he was staying in, an area that was highly trafficked by people and animals every single day.
Okay, wait. How do you possibly miss a plant with a leaf the size of a tree and a stalk as thick as a human thigh in your own backyard?
Because of a masterstroke of camouflage. Beccari had been walking past that exact spot for days without noticing anything extraordinary.
The petiole, which is the massive stalk that holds up that single giant leaf, perfectly mimics a lichen-covered tree trunk.
Really?
Yeah, Claudel's dissertation goes into great depth on this phenomena, calling it petiolar lichen mimicry.
Let me make sure I'm grasping the layers of this deception.
Yeah.
It spends a decade pretending to be in an edible, old, woody tree trunk just to avoid being eaten so that it can eventually gather enough energy to pretend to be a dead animal.
That's the long and short of it.
It's a master of disguise on multiple, completely different sensory levels.
It truly is. The stalk is biologically fleshy and green, but its surface is covered with numerous small orbicular white and pale green dots. This creates a visual pattern that perfectly replicates the cryptothecia or graphis types of crusty lichen you would see growing on the hard, old timber of surrounding trees. So it literally looks like old wood.
Exactly. Beccari literally brushed past it, assuming it was just another boring sapling. He only realized what it was upon very close inspection, noticing that the branching pattern of the canopy belonged to an aroid plant, not a woody tree.
I mean, it makes total evolutionary sense. A giant, fleshy, nutrient-rich stalk sitting in the middle of the jungle is basically an all-you-can-eat salad bar for herbivores.
Right, a very tempting target.
If it didn't disguise itself as hard, unappetizing wood, it would be devoured before it ever saved up enough energy in that corm to bloom.
Exactly the case. It's a vital, passive survival mechanism. But looking like a tree only keeps it safe while it grows. Looking and more importantly, smelling like a corpse is how it actually reproduces.
Which brings us to the main event, the phenomenon that makes this plant world famous. I have always wondered how we actually know the specific chemical makeup of a smell. I mean, did someone just hold a jar over the flower, bottle the stench, and take it back to a lab?
Well, the methodology is incredibly sophisticated. To understand the forensic chemistry of the stench, we turn to that comprehensive paper from Biochemical Systematics and Ecology. The researchers used a technique called gas chromatography mass spectrometry, or GCMS.
Okay, GCMS. How does that work in a jungle?
Essentially, they capture the air immediately surrounding the blooming flower using modified fibers. They're called headspace solid phase microextraction fibers. Think of these fibers as microscopic sponges designed to absorb specific airborne molecules.
Like soaking up the smell itself.
Right. Once they capture the scent, they run it through the mass spectrometer, which bombards the molecules with electrons, break them apart. By measuring the mass and charge of the fragments, the machine provides a highly precise chemical fingerprint of the air.
And what that fingerprint reveals is a cocktail of heavy sulfurous molecules that are a far cry from your typical sweet rose or lavender perfumes.
A very far cry.
Let's break down the actual recipe of this stench, because another study we're looking at, published in PNAS Nexus, actually mapped the metabolic pathways the plant uses to cook this up.
Yeah, the process starts on a cellular level with amino acids. The plant actively takes the amino acid methionine and metabolizes it into two highly potent volatile organic compounds.
And those are
Dimethyl disulfide, known as DMDS, and dimethyl trisulfide, or DMTS.
Okay, for you listening, if you want to know what DMDS and DMTS smell like, imagine taking a head of cabbage, throwing in a handful of crushed garlic, wrapping it in raw meat, and leaving it in a sealed plastic bag in the sun for a week.
That is painfully accurate.
That sharp sulfurous tang is the primary odorant responsible for the true carrion smell.
It forms the dense base layer of the scent profile. But the botanical alchemy doesn't stop there. The plant also converts another amino acid, arginine, into a compound called putrescine.
Oh man, the name alone is evocative. Putrescine.
Yeah, it's exactly what it sounds like. Putrescine is the literal chemical scent of decaying bodies. It is the exact same chemical produced when the amino acids in humans or animal muscle tissue begin to break down after death.
Wait, so the plant isn't just making something that smells kind of bad, it is synthesizing the exact chemical signature of biological death.
The precision of the chemical mimicry is staggering, and then the plant layers on additional accent notes to complete the illusion.
Accent notes, like a really gross perfume.
Exactly. The GCMS analysis picked up isovaleric acid, which produces a scent powerfully reminiscent of sweaty socks or rancid cheese.
Lovely.
It produces trimethylamine, which gives off an intense rotting fish or heavy ammoniacal odor. And finally, it produces indole, a compound that, in high concentrations, smells unequivocally like human feces.
I'm just imagining the sheer psychological assault of walking into a wall of garlic, rotting meat, sweaty socks, dead fish, and feces all at once.
An overwhelming sensory experience.
Because our human brains are hardwired by millions of years of evolution to feel intense disgust and nausea at those smells, right? Because they usually signal dangerous bacteria and disease.
Exactly.
But the plant backs up this chemical warfare with visual cues, too. It's creating a complete multisensory diorama of death. When that giant spathe, the frilly outer leaf, unfurls, the inside isn't the vibrant green of a healthy plant. It is colored a deep, mottled, dark burgundy.
The visual mimicry is just as crucial for the final stages of the deception. The biochemical systematics paper analyzed the non-volatile phytochemical constituents within the plant's tissues to figure out how it achieves that color.
What did they find?
Well, that deep, meaty, bruised purple color is rendered by the presence of massive amounts of anthocyanins concentrated in the external layers of the stade's tissues. Specifically, they identified cyanidin-3-O-glucoside.
So to a flying insect approaching from the canopy, it visually looks like a slab of torn, exposed muscle tissue, and it chemically smells like a decomposing carcass.
The illusion is perfect.
But Claudel's dissertation brings up a concept called odor polymorphism, which suggests the smell isn't just a static wall of stench, right?
Yeah, this is one of the most dynamic aspects of the bloom. The scent profile shifts and evolves continually throughout the night. The researchers noted that early in the bloom, as the spathe first begins to open, it might start with a slightly sweet, overripe, rotting fruit odor.
Like a rotting banana or something?
Exactly. Then, within the first 4 hours of full blooming, the concentration of dimethyl trisulfide and indole peaks. That is the intense rotting meat and dung phase.
Okay.
Later, as we move 12 to 17 hours past the initial bloom, dimethyl disulfide and trimethylamine reach their absolute maximum, shifting the scent profile dramatically toward rotting fish and pungent garlic.
That makes incredible sense from an evolutionary standpoint. I mean, it's casting the widest possible net. If a certain species of jungle beetle strongly prefers a 3-day-old carcass over a fresh one, or if a specific fly is drawn to dead fish over dead mammals, the plant eventually cycles through a scent profile that will trigger their specific instincts.
Right, it acts as a highly tuned, rotating broadcast tower, changing its frequency to summon different specific demographics of the insect world.
Which brings me to a detail I noticed in the GCMS data that completely threw me. Amidst all the rotting meat and fish compounds, the researchers detected a very specific chemical called 9-tricosine,
Yeah.
peaking right around that 17-hour mark, primarily around the actual tiny male and female flowers. Why is 9-tricosine so significant?
That is a brilliant observation, and it reveals a whole new layer of manipulation. 9-tricosine is not a byproduct of decay. It is a well-documented, highly specific sex pheromone used by several species of flies, most notably the common housefly.
Oh, wait, let me process that. So it is not just tricking them with the promise of food or a rotting place to lay their eggs. It's actively synthesizing an insect aphrodisiac. It's hacking their reproductive instincts directly.
It is multisensory, multi-layered behavioral hijacking. It exploits hunger, it exploits the instinct to lay eggs, and it exploits the mating drive. It uses every single biological lever available to pull these insects out of the sky and into its chamber.
This brings up a massive physical problem that the plant has to solve, though. We have established that it mixes this incredibly complex, heavy chemical cocktail.
Right.
But the Titan arum lives deep in the understory of the Sumatran jungle. The canopy above it is incredibly dense, meaning the air down on the forest floor is notoriously still.
Very stagnant air, yeah.
There is almost no wind to carry a scent. How on earth does a plant push these heavy sulfurous molecules miles into the dense forest to attract insects that might be far away?
To overcome that stagnant air, the Titan arum utilizes one of the most phenomenal adaptations in the entire plant kingdom. It turns itself into a furnace.
A furnace.
It undergoes a process called thermogenesis, meaning it literally generates its own intense metabolic heat.
See, when we think of warm-blooded organisms regulating their temperature, we immediately think of mammals and birds. We don't think of a plant running a high fever. But the data from the Plant Journal and the PNAS Nexus study is staggering. How hot are we talking?
The appendix, which is that tall, pale yellow protruding top section of the central spadix, can heat up to 12.6 degrees Celsius above the ambient temperature of the jungle.
Wow.
In a tropical environment, that means the plant is reaching nearly human body temperature, sometimes hitting the mid to high 30s Celsius, or roughly 98 degrees Fahrenheit.
To put your hand near it and feel actual radiating body heat coming off a flower must be incredibly eerie.
It really is.
And the functional reason it does this is to create what fluid dynamicists call a chimney effect. By drastically heating up the air immediately touching the surface of the spadix, that hot air rapidly expands and rises. It creates a powerful vertical updraft, like a localized weather system, that grabs those heavy, foul-smelling, volatile compounds and launches them high up into the rainforest canopy. Once the scent breaches the canopy, the stronger, faster air currents above the trees can catch it and carry the smell for miles. It's a biological smoke signal.
The physical mechanics of the chimney effect are elegant, but the biological how is even more fascinating. Because how does a plant, which doesn't have shivering muscles or brown fat like a mammal, spontaneously generate that much heat?
Right. How does it do it?
The PNAS Nexus study actually sequenced the transcriptome of the plant, meaning they looked at exactly which genes were being turned on and off to figure out the cellular machinery at work.
I know that normal cellular respiration involves breaking down sugars to make APP, which is the chemical energy currency of the cell. Think of a water wheel on a river. The water flows down, turns the wheel, and generates electricity. That's how most life functions.
That is the standard electron transport chain, yes. The Titan arum starts the same way. It pulls massive amounts of starch from that giant 150-kilogram underground corm we discussed earlier.
Right, the buried refrigerator.
Exactly. It converts that dense starch into a more mobile sugar, sucrose, transports it all the way up the massive, towering spadix, and then breaks it down into glucose within the tissues of the appendix.
But if it just turned that glucose into ATP, it wouldn't generate this massive burst of heat, would it?
No, it wouldn't. Here is the evolutionary trick. Instead of letting the water wheel generate electricity, the Titan arum utilizes a mitochondrial bypass. It expresses a massive amount of a specific enzyme called alternative oxidase, or AOX, within its cellular electron transport chain.
AOX.
Right. AOX essentially short-circuits the normal respiration process. It completely uncouples the flow of electrons from the synthesis of ATP.
So if we use an analogy, it's like taking a high-performance sports car engine, putting the transmission in neutral, and just slamming the gas pedal to the red line.
That's a great way to put it.
You're burning massive, unbelievable amounts of high-octane fuel, but the car isn't moving forward an inch. All of the energy from that combustion has nowhere to go, so it's released as pure, unadulterated, intense friction and heat radiating from the engine block. The The plant is burning through a decade's worth of painstakingly saved energy in a matter of hours, purely to run hot.
That analogy perfectly illustrates the sheer energetic violence of this bloom. It is a metabolic bonfire. This is precisely why the bloom can only happen once every 5 to 10 years, and why it only lasts 24 to 48 hours before the plant completely exhausts its energetic reserves, wilts, and collapses under its own weight.
But the deeper you dig into these sources, the more mysteries you uncover, because Cyril Claudel's dissertation actually throws a brilliant curveball into this whole thermogenesis theory.
Oh, the CO2 theory.
Yeah, the chimney effect makes perfect physical sense to spread the scent, but Claudel explores a compelling speculation about a secondary, perhaps equally important motive for all this metabolic burning.
It raises a profound biological question that researchers are actively debating. We call it the CO2 emission theory. When an organism undergoes massive, rapid metabolic burning of glucose, like our sports car revving in neutral drumpus, the natural, unavoidable chemical byproduct of that respiration is an enormous volume of carbon dioxide.
Wait, let me think about the target audience here. Carrion insects, flies, and beetles don't just use their sense of smell to find decomposing matter, do they? They also track high concentrations of CO2.
Exactly. A freshly dead animal doesn't smell immediately. But the millions of microbes and bacteria that immediately start breaking down that carcass undergo rapid respiration, exhaling a huge plume of carbon dioxide.
Oh, wow.
Carrion flies are hardwired to follow CO2 gradients to find fresh graves, so to speak.
But I want to push back on this a little, because biology can be messy. Couldn't the massive CO2 output just be a total coincidence? If the plant's primary goal is to burn glucose to make heat for the chimney effect, massive CO2 production is just the inevitable biological exhaust pipe. How do we know the CO2 is an intentional evolutionary trap rather than just a side effect?
That is the exact crux of the debate within the botanical community. Is it a happy accident or an actively selected trait? While we can't definitively prove intent, evolutionary biologists point to the fact that the Titan arum tightly regulates when this burning happens.
The timing is
very specific. It synchronizes the peak heat and therefore the peak CO2 exhalation exactly with the opening of the spathe and the release of the primary carrion scents. It acts as a highly synergistic effect.
I see.
The heat creates the updraft to spread the VOCs, while the simultaneous massive exhalation of CO2 provides an additional, highly specific chemical cue that convinces the flies they are approaching a breathing, living mass of decomposing bacteria.
It just highlights how flawlessly this plant mimics a corpse on every conceivable sensory level. Visual color, volatile scent molecules, physical heat, and even the breath of bacterial decay.
It's unparalleled mimicry.
Which brings us to the actual victims of this elaborate deception. Because all this burning of years of stored energy, all the complex sulfur chemistry, all the heat and CO2 is aimed at a highly specific audience. Who exactly is answering this foul beacon in the middle of the dense Sumatran night?
As we've established, this plant has zero interest in bees, butterflies, or hummingbirds. It targets nature's undertakers. The primary visitors answering the call are carrion beetles from the family Sylphidae and flesh flies from the family Sarcophagidae.
Right, the undertakers.
The biochemical systematics paper specifically noted the heavy presence of blue bottle flies and blowflies, which you can easily distinguish by their bright, iridescent metallic blue and green exoskeleton.
Oh, those shiny ones you always see on garbage.
Exactly those. Interestingly, Claudel's field research also documented certain species of moths visiting the bloom during the night, which hadn't been widely integrated into previous pollination models.
So these insects follow the scent plume, they follow the heat, they follow the CO2, and they finally arrive at the source. But what happens when they get there? Because this isn't a normal pollination scenario where a fuzzy bee lands on a daisy, gets a nice sugary sip of nectar for its trouble, and happily flies away.
Far from it. This is a phenomenon known as oviposition site mimicry.
Oviposition, meaning egg-laying.
Exactly. The deception reaches its climax here. The plant is deceiving these insects into believing this fleshy red chamber is a prime location to lay their eggs, their oviposition site. The flies and beetles dive deep into the dark, warm, stinking chamber created by the spathe, heading straight down toward the base of the central column, where the actual microscopic male and female flowers live.
Let's walk through the physical mechanics of this trap because it is diabolical. To explain it, imagine being invited to a gorgeous, fully furnished model home. You walk in, the door locks behind you, the floors are covered in grease, and you slowly realize all the fruit in the kitchen is made of plastic and there is no plumbing.
It's a house of horrors for a fly.
The insects fly into the spathe, But the interior walls are incredibly slick, almost waxy. Furthermore, the base of the central spadix, the appendix, has a distinct overhanging wall, almost like a botanical retaining wall.
Once the insects slip and slide down to the very bottom of the floral chamber, they find themselves in a dark, confined space. They stumble around frantically in the dark, bumping into the floral structures, and when they try to leave, they find it incredibly difficult to climb back out over the slick walls and that physical overhang.
They can't get out.
No, they are effectively trapped in a holding cell.
And this is where the sheer brilliance of the plant's timing comes into play. It uses a mechanism called protogenous flowering.
Yes. The plant intentionally separates its gender phases by time. This is a crucial adaptation to prevent self-pollination, which would lead to genetic inbreeding.
Okay, so how does that work with the trapped flies?
On the first night of the bloom, when the stench, the heat, and the CO2 are at their absolute overwhelming peak, only the female flowers, which are situated at the very bottom of the chamber, are biologically receptive.
So picture these trapped flies. They are thrashing around in a panic in the dark, desperately trying to find the rotting meat they smell so they can lay their eggs. In their chaotic scrambling, they are inadvertently brushing up against the receptive female flowers, depositing any pollen they might have brought with them from another Titan Arum they visited a few days prior.
They execute the crucial cross-pollination work on night one, but the plant isn't done with them. It keeps them trapped in the chamber overnight.
That is just wow.
It isn't until the second day, usually around 24 hours after the initial opening, that the male flowers, which sit in a ring just above the female flowers, finally mature and open. They begin to shed massive amounts of sticky pollen, showering the trapped, exhausted insects below in a fresh, heavy coating.
And only then, once the flies are thoroughly coated in the new genetic material, does the plant let them go. The slick inner walls of the spathe begin to wither and lose their waxiness. That overhanging wall shrivels up, breaking the physical confines of the trap and releasing the bugs to fly off into the jungle, completely exhausted, only to be fooled by another corpse flower miles away.
It is a perfectly choreographed, tightly controlled sequence of physical and chemical events.
But looking at it from the insect's perspective, this is incredibly one-sided. In normal mutualistic pollination, it's an economic trade. The bee provides a transport service, and the plant pays the bee with high-energy nectar. Here, the flies get absolutely nothing.
Nothing at all.
They get physically trapped, they expend massive amounts of energy trying to escape, and worst of all, they are duped into laying their eggs inside a flower. The biochemical systematic study explicitly noted that researchers found hundreds of fly eggs and wriggling larvae scattered among the male and female flowers at the base of the chamber.
Yeah, that's a grim detail.
Those larvae are going to hatch expecting a nutrient-rich rotting carcass, and they're going to starve to death within days because there is no actual meat to consume. It's pure, unadulterated exploitation.
It is the dark side of botany. It's a masterpiece of evolutionary manipulation. The plant invests an astronomical amount of energy into creating the flawless illusion of a reward, rather than expending energy to provide an actual reward like nectar.
It's ruthless.
It is a high-risk, high-reward strategy that has allowed it to secure highly specific, dedicated pollinators in the intensely competitive, chaotic environment of the dense rainforest.
This incredibly specific, complex biological trap is a marvel of millions of years of evolution. It is perfectly, flawlessly tuned to its exact environment. But that brings us to the tragedy of the Titan Arum. Because it relies entirely on a few specific species of flies, a perfectly calibrated jungle temperature, and highly specific humidity levels, it's incredibly fragile. What happens when that perfect jungle environment starts disappearing?
This is where we must shift our focus to the stark conservation status of this giant, because the reality on the ground is sobering. Despite its monstrous size, its fearsome appearance, and its aggressive chemical warfare, the Titan Arum is profoundly endangered.
Just how endangered is it?
According to the latest data and the IUCN Red List assessments, estimates suggest there are only around 303 mature, reproducing individuals left in their natural habitat in Sumatra.
303. That number stalks you in your tracks. 303 individual mature plants left in the wild on the entire planet. Why is the population collapsing so rapidly?
The primary driver, which echoes the crisis facing countless species across Indonesia, is severe, unchecked habitat loss. The Titan Arum is not an adaptable weed. It relies on incredibly stable, specific microclimatic conditions.
Right.
According to the comprehensive 2024 field study by Setyawan and his colleagues, which analyzed optimal habitat profiles, the plant strictly requires ambient temperatures between 20 and 30 degrees Celsius, humidity consistently hovering over 80%, well-drained volcanic soil rich in decaying humus, and the deep protective indirect shade provided by a fully closed primary forest canopy.
And those exact delicate conditions are vanishing at an unprecedented rate. Sumatra is experiencing rapid, large-scale deforestation. The primary driver of this clearing is the massive expansion of agro-industrial enterprises, specifically monoculture oil palm plantations.
Yeah, the oil palm industry.
When you bring in the bulldozers and clear the ancient canopy, the entire microclimate shatters. The humidity plummets, the ground temperature spikes under the direct sun, the rich topsoil rapidly degrades and washes away, and the Titan Arum simply cannot survive the shock.
The destruction of the canopy is a death sentence for the species. But the Setyawan paper also highlights that it's not just massive industrial logging causing the decline. There are deeply localized, culturally complex human threats that are actively exacerbating the problem.
Like what?
Well, for example, in the Kapayang Regency of Sumatra, researchers found that locals sometimes actively seek out, harvest, and destroy the plant due to persistent folklore. They cut open the massive petiole, searching for a mythical sacred stone, or talisman believed to be hidden inside.
The sacred stone.
Yes, which is locally thought to possess mystical properties or cure severe diseases.
It is heartbreaking. They're cutting down a plant that spent 10 years quietly saving energy, killing it before it can ever reproduce, looking for a magical stone that biologically does not exist.
It's a tragedy of misinformation. Furthermore, remember that incredible lichen mimicry we spent so much time discussing earlier? The mottled grayish-green, spotted pattern on the giant stalk that protects it from herbivores.
Yeah, the camouflage.
Well, to many locals walking through the dense underbrush, that specific pattern looks uncomfortably similar to the scaled skin of a large python or a venomous snake.
Oh.
Out of sudden fear and a startled reflex, they will often slash and destroy the plant with a machete when they encounter it.
Talk about an evolutionary backfire. The very camouflage pattern it evolved over millions of years to protect itself from wild animals is now the exact trait getting it killed by human beings.
Exactly.
And I think there's another insidious modern problem here, which the conservation literature indirectly points out. If you open YouTube right now and search for corpse flower, you will find dozens of high-definition time-lapse videos of Titan Arums blooming in prestigious botanical gardens all over the world, from Kew Gardens in London to the U.S. Botanic Garden in Washington, D.C.
Well, they're wildly popular online.
These videos have millions of views, but it creates a psychological illusion. It makes the plants seem ubiquitous, like they are everywhere, thriving and doing just fine. It completely obscures the grim, silent reality of their rapid slide toward extinction in the wild.
It creates a very dangerous false sense of security among the public. What you're describing is ex situ conservation, meaning keeping and protecting the plants outside of their natural habitat in controlled botanical gardens. Ex situ conservation is undeniably vital for public education and raising awareness.
But there's a catch.
A big one. The Setyawan study notes that there are currently about 500 captive Titan Arums documented globally. But there is a critical hidden biological flaw in relying on this. The genetic diversity of those 500 captive plants is dangerously low.
I see.
Many of them are closely related, grown from seeds collected during the same few historical expeditions. If a novel pathogen or a specific fungal disease sweeps through those greenhouses, the entire captive populations will be wiped out because they lack the genetic variation to resist it.
It's terrifying.
If the long-term true survival of the species, we absolutely need in situ conservation. We have to save them and their genetic diversity in the wild forests of Sumatra.
And that is exactly where the 2024 work by Setyawan and his team in Indonesia provides a massive glimmer of hope in what is otherwise a dark story. They aren't just documenting the decline and watching the plant go extinct. They are actively, aggressively fighting to save it, using advanced botanical technology. They're undertaking large-scale in vitro propagation.
The methodology they're using is fascinating, and it's a testament to human ingenuity. Because wild seeds are incredibly rare, remember, successful cross-pollination requires two of these extremely rare plants to bloom at the exact same 48-hour window, relatively close to each other, with the right flies present. The researchers can't just go collect seeds.
So what do they do instead?
They're cloning them. They carefully take tiny microscopic tissue samples called explants directly from the living petiole of a wild plant. They bring these tissues into a highly sterile lab environment and treat them with a highly calibrated cocktail of synthetic growth hormones, specifically BAP and NAA.
Okay, for those of us who aren't cellular biologists, how do BAP and NAA actually trick a piece of stem tissue into becoming a whole new plant?
It is essentially hacking the plant's cellular programming. BAP, or benzolaminopirin, is a synthetic cytokinin. Its primary job is to aggressively stimulate cell division. It forces the plant cells to rapidly multiply, creating a mass of undifferentiated tissue called a callus.
Okay, so a blob of cells.
Right. Then they introduce NAA, or 1-naphthalene acetic acid, which is an auxin. Auxins specifically signal the cells to begin differentiating and pushing out roots. By perfectly balancing these two hormones, the researchers convince a tiny scraping of stalk tissue to forget it was part of a stalk and instead organize itself into a brand new, genetically identical, independent plantlet with shoots and roots.
It is botanical science fiction happening in real time, and the most inspiring part is that it is actually working. They successfully grew these tiny clones into viable, healthy seedlings in the lab, but they didn't stop there. They aren't just keeping them locked in a sterile vault. They are actively reintroducing these lab-grown seedlings back into the wild terrain.
Yes, that's the crucial step.
The Setuan study detailed how they successfully planted 100 hardened seedlings in the Teletang-Kamang district of the Igam Regency and another 20 in the Payo, Tanagarm district of Solok City.
The scientific achievement is monumental. But what is perhaps even more crucial about the specific reintroduction effort is the sociological approach. They are actively involving the local communities in the planting and monitoring process.
That's amazing.
They're holding educational workshops, explaining the unique biology of the plant, dispelling the harmful myths about the sacred medicinal stones, and clarifying that the snake-like pattern is harmless camouflage. They're working to shift the cultural narrative, trying to turn the Titan Arum into a symbol of intense local pride, a flagship species for conservation, and a potential driver for sustainable ecotourism, rather than an object of fear, superstition, or exploitation.
It really makes you pause and marvel at the sheer stubborn tenacity required for modern conservation. You have dedicated botanists hiking for days into the sweltering, dense, leech-infested jungle, tracking down a faint rotting smell on the wind. They carefully, surgically cultured the microscopic tissues of this bizarre, giant, alien-looking organism in a high-tech lab, nurse those fragile clones for months, and then hike back out into the mud to plant them in the ground.
It's an immense amount of labor.
All of this immense effort just to ensure that this incredible, foul-smelling botanical oddity doesn't quietly vanish from the earth forever.
If we step back and connect all these wildly different threads, the volatile chemistry, the heat generation, the insect manipulation, the deforestation, it leaves us with a rather profound and sobering biological concept to consider.
What's that?
Think about the intricate, invisible, ancient interconnectedness of the rainforest. The Titan Arum has spent millions of years perfectly tuning its chemical vocabulary. It has painstakingly dialed in the exact microscopic ratio of dimethyl trisulfide to mimic death. It has calibrated the exact cellular heat required to create an updraft in stagnant air. It has perfected the exact synthesis and timing of the 9-trixene sex pheromone.
Yeah.
It has learned to speak a highly specific, flawlessly articulated chemical language that only a tiny handful of specific carrion flies and beetles can understand.
It is a lock and a key, shaped by deep time.
But if those specific insect populations decline or disappear entirely due to the exact same habitat loss and microclimate collapse that threatens the plant, the Titan Arum is left desperately screaming its complex chemical signals into an empty void. It becomes an organism perfectly, beautifully adapted for a world that simply no longer exists. It begs an incredibly important question for us to ponder long after this deep dive ends. What other incredibly complex, invisible chemical conversations are happening right now in the undisturbed dark corners of the world? And what are the catastrophic cascading effects when human activity accidentally silences one half of that dialogue?
That is a haunting, beautiful, and urgent thought to end on. You don't just lose a single giant plant. You lose an entire ancient, invisible conversation that has been echoing through the jungle for millions of years. Thank you so much for joining us in the dense, stinking undergrowth of Sumatra today for this discussion. We hope you stay insanely curious, keep questioning the bizarre and brilliant mechanisms of the world around you, and remember that sometimes the most miraculous, perfectly engineered things in nature are the ones that make you want to plug your nose and run the other way.